Welcome to the podcast on cell and gene therapy. And I'm excited to have a conversation with Dr. Pratima, who is the General Manager and Senior Vice President of BioNova based out of Indianapolis. She has a PhD in biochemistry and molecular biology and also an MBA from Indiana University. So she has both the science and the business aspects of it. She's been chief scientific officer of other companies and has a lot of experience in the field of cell and gene therapy. So welcome to the podcast, Pratima.
Thank you, Nari.
I'm happy to invite Pratima, who has written the chapter with her colleagues on cell therapy. But I'll also tell you that she's also equally an expert in another chapter, which is gene therapy, which a lot of her colleagues actually had contributed to writing. So she was toggling between both chapters. So I thought, Pratima, today we'll talk about both cell and gene therapy, which is sort of the expertise that you have, and also see where the field is going. So welcome to the podcast.
Thanks, Nari. Thanks for having me. And also thanks for giving me the opportunity to be a part of this book. It was wonderful to work with everyone and the support that we got from you and how you managed. There were a lot of people that you were
A
managing, so
lot
it
of
was pretty
moving parts.
impressive. Thank you.
No, and everybody worked together. Thank you so much. So, as an introduction, Pratima comes with a lot of experience. She has a PhD in biochemistry and molecular biology. She has an MBA to top it. So, a lot of experience in not just in the science, but also the business aspects of drug development in general. She's been chief scientific officer for a few companies, but currently she's chief scientific officer of a company called BioNova, and you're based in Indianapolis. The article talks about the diversity of topics that you've talked in the cell therapy. So again, let's spend some time talking a little bit about the field, and then we'll talk about the book itself in the second half. So I guess my first question is if you can give a little bit of an overview to the viewers of the podcast as to where the cell therapy, so a little bit of the history of the cell therapy and where it is going, so a very high level, because it, for many of the readers or authors, cell therapy may be a new subject.
Sure. Yeah. I'll try my best. Most of, I don't know, people who are listening to that, some of them might know where it started and some of them might not. But the thing that surprises most people is how new this field is compared to your small molecules or the antibody or the MABs field, right? It's still about maybe 20 years old, really. That's about it, right?
Right.
Um, the first cell therapy, I would say, which was when cell and gene therapy is not as famous as today, it started with a retroviral vector.
Mm,
That's for ADS kid, right? And it was very promising when it was tried, but unfortunately, retrovirus also had this pro-insertional what's it like it's pro-oncogenesis, like insertions happen in before the promoters of oncogenes.
mm,
Unfortunately, because of
mm,
that, there was a death of a child that was involved in the clinical trials, and then it got to a halt. But since
mm,
then, with LVV, invention of lentiviral vectors, there are more safer vectors that has been developed, which means that when the recombination happens, the virus get inactivated, right? So
right.
with that, what we see today is of all the drugs that have been approved in cell and gene therapy, the safest drugs so far that are staying in the market are the cell therapy drugs,
Wow.
right? The
Wow.
ones approved. And
Mm,
then going forward, actually, the field is getting really interesting, Naren, that there
mm,
is in vivo cell therapy that people
mm,
are starting to move into, right? So it's activating your T cells or your stem cells with vectors, and then also expansion within the body itself. So
right.
I think that might be where everybody would move eventually.
Yeah. With cell therapy, obviously with bone marrow transplant, there are probably hundreds of hospitals in the United States and all over the world which have the potential to do bone marrow transplants. Do you think these centers that do bone marrow transplants are conducive to do this kind of therapy? And what would they need to be able to convert their, I would call not simple, but the bone marrow transplant unit to a cell therapy unit?
I think it's mostly the labor that is experienced
Mm,
in your cell manufacturing, right?
mm,
Especially if it's autologous therapy,
mm,
you need to be, yes, they can isolate cells, your panty cells from the patients, but they would need to have a whole unit, right,
mm,
where they
mm,
can isolate the cells, they can expand the cells, they
mm.
use viral vectors or other type of vectors that can introduce the gene of interest and then grow them up and then
Yeah.
the analytics that are needed before they can put back in the patient, right? I think that the little ecosystem needs to be there for the
Mm.
hospitals or the doctors that want to do cell therapy in hospitals.
Okay.
Now, there are few, I'll have to remember the names, but there are few companies that are trying to build that these like cocoon is
Mm,
one of the cell manufacturing equipment, right? But they're building these small GMP units,
mm, mm,
which can incorporate these equipment,
mm.
and then they also will provide 1 or 2 FTE to be at the hospital to do this. There are a few companies that are starting these. I can't remember off the top
Yeah,
of my.
but the idea is
The
that
idea
these
has,
companies, yeah,
yeah,
yeah, yeah.
yeah.
Now that seems like an interesting proposition, especially if you have to go to countries and regions where all these resources are not available.
Yeah.
Switching gears a little bit to from cell therapy to gene therapy, the field of gene therapy is depends tremendously on these viral vectors today. One of the challenges in the field has always been access and availability of these gene therapy vectors for people. So, can you tell us what the challenges are from a manufacturer's perspective uh, of these viral vectors?
Yeah. As we discussed before, it's still a very new field, which means that during manufacturing and development, right? If it is cell therapy, I feel like that might be a little easier because you have lentiviral vectors with
Mm,
VSVG and all of that. That'll pretty much transduce any cell type.
I
So I
see.
think that's kind of easier. That's the easier part, I would say, for the cell therapy. For the gene therapy, unfortunately, when you're thinking about AAV, specific capsids, the specific serotypes that you need to use to get to
Mm,
the cell type that you need, and that's becoming a little tricky. Plus, the cost of goods due to the youngness of the field and the manufacturing processes are not there as your maps are,
mm,
unfortunately.
mm, mm.
The recovery, be it whatever type of vector, even the best of the best manufacturing process will only give you about 35% recovery at the end. So
Wow,
the
wow,
loss,
wow,
yeah.
loss is
So
huge.
the accessibility unfortunately starts with your development and then your initial clinical trial manufacturing processes, right? If your recoveries are so low and the final product that you make, most of it is gone for your analytics, the backups,
Yeah.
retails. Uh, so, so better analytics and then also better recoveries would
Recovery.
definitely increase the
In,
accessibility.
so
Yeah.
that's in your mind. That's where the innovation has to happen to
Yeah.
improve COGS and yield. Yeah, yeah. No, excellent points. So switching gears a little bit and talking about the chapter, I think as most of the people I've talked to in this chapter, including myself, we generally don't write chapters of textbooks when we publish. So, what was your experience in writing the chapter, and was it what you how was that?
I was, as you know, I was collaborating with many others, right?
Yes,
My
yes.
focus was definitely on the viral vector production for like how you design the vectors for your cell therapy, the design of the manufacturing process itself,
Hmm.
and what you need to think in terms of quality,
Yeah.
right? Like starting with quality by design in mind is
Yes.
what would be helpful is what I've contributed to mostly. Now, when it comes to working together, I mean, these are all brilliant people now, and like the people that you've put together, what they've done is brilliant, and I added my little part to that. The help that I got, especially in terms of um, I wrote it, but then it has to be in the format for presentation for the
Sure.
text, right?
Sure.
And then the diagrams, the ideas were mine, but you've people who really help them to unify the look and the format of the textbook. I think that has been really a great help. Like brilliant people, good support. This is actually my second chapter.
Wow. Okay.
Yeah, like being an author. Yeah. The first one was Alliance for Regenerative Medicine. This was in 2023.
Okay.
Uh,
Okay.
yeah,
Yeah,
it's been a pleasure, I would say.
you've had experiences, yeah. So I think, as you mentioned, writing a chapter is quite different from writing a manuscript, where we have much more control over what we're writing. Um, So, on next question, I think is related to more career development. So, this book is targeted at young professionals, students, masters, PhD students who are going to read this book. So, from your perspective, what is your advice of this field of cell and gene therapy as a prospect of the future in drug development?
Yeah. For young professionals who are using this, I would say they should use this as a tool to grow and to grow their ideas and build on what's already there at the ground
Mm.
level, right? I would take this book as like a ground level, like the basic principles of what's available as of today. As the field grows, regulations change, and then also new technologies and needs to emerge, right? Again, like I said, recovery is the major roadblock, unfortunately, as of today. If
Mm.
these young minds can develop a process where we don't have to use as many columns, for example,
Yeah.
that itself will remove about 45% of your cost of goods.
Mm.
So ideas there to make the viruses, because most of them start as of today, still it's viruses, right? 90% I think is viruses. There are LNPs and mRNA that are the new fields coming up.
Correct.
That'll be really helpful for the field, knowing like these young minds coming up with new manufacturing processes. I think that's what I'm looking forward to from people.
Yeah, no, I think yeah. So I think this is an exciting field, and I keep telling myself as I read these, I wish I was 50 years younger.
Yeah, that's true.
But it's such an exciting field, and the field of data analytics and
Yeah,
biology is all coming together.
that's true.
So maybe in that regard, I'll ask you a question. Are you using, or is the field using machine learning and AI kind of tools for this kind of technology?
I'll be very honest with you. I personally haven't used it, but I know in my new role here, um, so I'm upstream of viral vector. No, in my new role, it's plasmids.
Hmm.
But I'm pretty sure what I'm being introduced to and what I'm also exploring are something called digital twins. So
Oh, right.
the DOI studies can be, I'm actually working with this company called Data How, and then there's another one that sorry, Kasei Bioprocess Unit owns. These are the two companies that I'm exploring as of today.
Fantastic.
So they'll take what they do is they take you'll have to have basic data, right? Once you
Yes.
have a couple of runs under your belt, it's going to take all the process parameters and then apply it to your new therapeutic gene. Let's say after my 20 runs of different plasmids and different viral vectors, that data can be applied so that my DOE studies initially can be
Hmm.
reduced, or they can be very well defined that I know what the output is going to be. right?
Mm,
It's
mm,
definitely going to help that way.
mm.
Uh, that's my understanding, and that's what I'm trying to explore
Yeah,
in my
yeah.
role here.
Yeah, I think it's so exciting to be part of this process of all technology is so exploding
Yeah,
for some reason,
yeah,
right? But there's
yeah.
something happening in the world
I
where
know,
all of these
I know.
technologies are coming together, and
Yeah,
you just have to learn each of these
yeah,
technologies.
yeah.
Yeah, and it's been exciting to sort of put this book together at this time. However, I feel like it's been 6 months or so now that the book has been published or will be published, and it seems like it's already out of date with
Yeah,
so
yeah.
many things that are being published.
That's how it feels. I know, seriously.
You need to
But
keep
I'm glad you guys are doing these
podcasts. Yeah,
podcasts. I think that helps keep up the innovations up to date.
yes, yes, yes. So yeah, so we wanted to keep this brief discussion also because it allows people to see a face to the name when they're reading the textbook and understand what concepts came up. So thank you very much for taking the time to chat with me a little bit about the textbook and the field in general. For all of you viewers, this is a continuing podcast that we are doing on each of these chapters on this book, Approved Life Cycle of Drug Development through Springer. And we look forward to you joining our next episode. So thank you very much, Pratima.